I'm David Woodland. I'm the editor-in-chief of the Journal of Interferon and Cytokine Research. I'm here with renowned viral immunologists Paul Thomas and Katherine Kedzierska, organizers of the Keystone Symposium meeting on viral immunity, basic mechanisms and therapeutic applications, which will be held in Keystone, Colorado on January the 17th to the 20th, and also live streamed virtually. Welcome. Viruses infect millions of people around the world, causing severe illness and straining public health services. The Keystone Symposium meeting on viral immunity will bring together leading scientists in the field to present and discuss cutting-edge research in the field. What was your vision for organizing this meeting, and how does it stand out from others in the field?
So I think it's important to let everyone know we started planning this meeting in 2019, long before we knew there was going to be a coronavirus pandemic. And our vision then was to really capture all aspects of viral immunity and bring together leaders that could talk about the major questions that are still outstanding in the field. And so this includes a broad spectrum of viruses infecting different tissues, and different lifestyles of those viruses, as well as different arms of the immune response. So everything from very basic mechanisms of innate immunity all the way through adaptive immunity and the development of therapeutics that exploit or promote all of those antiviral activities.
I'd like to say that we are immensely excited about this upcoming Keystone meeting on viral immunity. Personally, I've been attending Keystone meetings since 1998, since I started my PhD. And for me, Keystone meetings are always associated with this cutting edge, hot of the press data sharing, this vibrant energy in the room, stimulating discussions during the poster sessions or even later at the fireplace until long hours in the evening. So yeah, as Paul said, our main goal is to foster these multidisciplinary collaborations across different approaches, different viral models and outcomes, and really to provide a unique opportunity for interactions. We really feel that this Keystone Symposium will reconnect viral immunologists so they can share the data in this rapidly changing field.
And I think the cross-fertilization piece is really a wonderful aspect of Keystone Symposia conferences.
Again, from the beginning, this meeting was designed to be co-run with the respiratory viruses meeting, and so it's a wonderful synergy. And obviously, now a lot of the work is focused on the impressive accomplishments that have been made in the field around coronavirus. And so there's a parallel meeting on respiratory viruses with some shared plenary sessions that will cover not just coronavirus, but also flu, RSV, and others. And it'll just bring together even larger groups of people, whereas our meeting has viruses beyond respiratory viruses and a real focus on immunity. The respiratory virus meeting is more of a pure virology meeting, and so also contains talks that are more mechanistic about viral replication and lung damage and repair. And so I think, it's really going to be what, as Katherine said, Keystone is best at, bringing together diverse groups of scientists to interact with each other. And this, especially after this period where we've been virtual for so long, having people in person to share the experience of the last year and a half, both personally and scientifically, will be very valuable.
Yeah, I think it'll be definitely exciting to get people back together in person again. I think everyone's really anxious for that to happen. What do you think the most exciting new advances and research directions that will be covered in the program?
So for me, I would say it's human immunology, really providing an in-depth understanding of how our immune system works to recover from viral infections and how immune system is perturbed in high-risk groups. As Paul said, when we started planning this meeting in 2019, we've had one session on human immunology. However, with the recent discoveries in human immunology, with an outbreak of SARS-CoV-2, in understanding human immunity to viruses in blood tissues, I really think we'll see human immunology throughout all the sessions and the recent advances.
So many viruses, including SARS-CoV-2 and influenza, induce hypercytokemia. So what do we know about the mechanism of induction and its impact on the course of infection and/or clinical symptoms and severity of disease?
So it's a great question, and it's been a major focus of the work in SARS-CoV-2 in part because of the success of the therapeutics targeting inflammation in very severe cases. I think what what's emerged is a picture that the innate immune response plays a major role in the early induction of hypercytokemia, and the innate response in recognizing specific aspects of the individual viruses. So I think, the cytokine storm in SARS-CoV-2 is potentially not the same cytokine storm that we see in certain influenza virus cases. And so it's really that, viral host interplay that's driving that response. On the host side, I think it's very specific host factors that lead to that particular phenotype. And I think it's a fairly exceptional phenotype. So while it is a significant proportion of the groups that suffer very severe disease, I think there are many different ways to suffer a severe outcome from any viral infection, but including these respiratory viruses. And so figuring out which ones are overinflammation, underinflammation, or the wrong kind of inflammation is something that the field is still really grappling with. And that's going to have to drive much more targeted therapeutics.
Yeah, so to add to this, the way I see it is it is a race between our immunity and the virus. And the earlier our immunity kicks in, then the sooner we can control the virus replication, pathology. However, delayed immune responses, as we see in influenza patients, COVID-19 patients, lead to obviously more virus replication, more pathology, and that's getting out of control. And that's when the virus, when the immunity gets initiated, it leads to hyperactivation of the cellular components, hypercytokemia. And this is why the vaccines are so powerful, because we generate immunological memory that can initiate those early immune responses after infection, so we can win the race with the virus.
Is hypercytokemia always present in severe cases, or is it only in some cases of COVID?
I think it's still a controversial area. And our own work in SARS-CoV-2, we found it actually in only a minority of severe cases, but a significant minority. I think one issue is with the timing of when samples can be acquired and where samples can be acquired. So you're going to get a different story from the blood than you are from the mucosal surfaces. And the onset of various therapeutics has also complicated this. So if we're sampling people after they've been treated, then we're not getting an understanding of precisely what the immune response was doing in the absence of that intervention. And so in the same way for flu, we know in some very severe flu infections, H5 and H7, they're often characterized by hypercytokemia, and that's some beautiful work from Katherine's lab that has shown us that. But there's seasonal flu severe cases where people appear immune suppressed. And it's the same story for SARS-CoV-2, where the immune system actually seems to be downregulated from even a mild infection. And again, that could be a timing issue as far as are we seeing some counter-regulatory effects of the immune response having geared up too high and then trying to shut itself down to avoid the pathological outcomes.
Is it the case that this hypercytokemia is different in flu versus SARS-CoV-2 infections? And what is that difference, if so?
So the individual cytokines that are upregulated are different, and the kinetics of when they're upregulated are different. And I think these are high parameter data sets often built on relatively small numbers of patients in any one study. And so this is, again, something I think where the field really has to come together to pool data and really do some meta-analyses to see what the underlying differences might be and the underlying etiological differences. One very sort of stark case where there's a clear difference is the difference between MISC patients, which develop this, what is clearly a hypercytokinemia, but after the clearance of the virus. And so these are, kids that have the severe inflammatory disorder but can generally be rapidly treated with IVIG and steroids and recover in almost every case, versus the hypercytokinemia in elderly individuals that develop in the middle of the infection and often respond to immunomodulatory therapeutics, but not always.
What is the status of research into the role of interferons in the immune response to SARS-CoV-2 infection?
With coronaviruses more broadly, there's a sense that compared to other very well-studied viruses, especially influenza virus, which has driven a lot of our thinking about respiratory infections, that the coronaviruses have a lot of tools available to them to avoid the type 1 interferon response. So they have, it's a much larger genome, and they have many dedicated gene products that are specifically attacking type 1 interferon induction and the effects of specific ISGs. So at baseline, the viruses are already maybe, as a group, coronaviruses are better at evading interferon responses. And then there's, a series of very interesting findings that individuals that suffer severe SARS-CoV-2 may be more likely to have defects in type 1 interferon pathways, genetic defects, either in the sensors that, the innate sensors that induce type 1 interferon, or in type 1 interferon itself and its receptors. And then lastly, there's the work that suggests that some individuals mount an autoimmune response to the interferon proteins themselves, and as measurable by antibodies directed against the type 1 interferons. And again, piecing together what proportion of different severe outcomes are contributed to by each of these mechanisms, I think is still a really key element. And then how therapeutically to both detect these early and then intervene is another major question.
What are the symptoms of this autoimmune syndrome?
Well, so it's a subset of the people that had very severe disease. So that's how it was identified. I think the original paper was out of Rockefeller, and it was with Casanova, and he's always looking for genetic factors. And so he had accumulated this huge set of people with really poor outcomes. And I think maybe specifically people that weren't otherwise, obvious high risk. So, these were maybe a little bit on the younger end. Mm-hmm. And rather than, he has found some genetic factors, but I think in terms of like the single largest feature, it's this response. But that said, it's all post the development of severe disease. So it would be great to know what those people looked like 6 months before, and whether or not there was any sign that they developed that. The papers that have come out so far, they were sort of shocking, right? So there's a good history of viruses inducing kind of off-target antibody responses. And so I think that's why these groups were even looking at it to begin with, because it was sort of a weird finding, but in multiple groups found it sort of pretty quickly simultaneously. So I think they were doing sort of broad antibody arrays. But it seems to be a consistent finding where it's not just that you all of a sudden start making antibodies to everything in your blood. It's very interferon focused. But it's only been something that I would say is recognized in the last year or so. And nothing else is circulating. So now we know those people exist, whether or not there's long-term consequences for them, whether it was SARS-CoV-2 specific, and whether or not this is the kind of thing that happens in other viruses, I think is still kind of unknown.
Yeah.
I think SARS-CoV-2 is just, I love flu, and I will never leave flu, but I think SARS-CoV-2 is a much more sophisticated virus than flu. It just has so many more tools. And so I do kind of wonder if it's something that's a consequence of all its manipulation of the interferon pathway.
Catherine, do you have anything to add?
Just that the clinical trials show promising results with the administration of interferon type 1 in COVID patients, and can reduce hospital stay and mortality, and prevent immunopathology.
Okay. So the next question is something I'm particularly interested in, and that is that many viruses enter the body through mucosal surfaces. So what do you consider to be the unique aspects of immunity at mucosal surfaces, and what are the challenges for vaccination?
So mucosal immunity is obviously of a great importance, especially for respiratory viruses, as this is where the viruses are detected, for example, by toll-like receptors, critical in early detection of viruses, activating innate immunity, then bringing in the adaptive immunity. So one challenge is we still need to understand a great deal about the mucosal immunity. However, with SARS-CoV-2, there have been a number of beautiful studies on understanding immune responses at the mucosal sites, and not only in the blood, but also in tissues. One challenge is obviously the delivery of the majority of vaccines for respiratory viruses that's not generally target a great deal of mucosal sites. And on top of it, we are still measuring antibodies in the blood after for those viral infections. So really a broader knowledge of antibodies, innate immunity, adaptive immunity at the site, at the mucosal surfaces is needed. And so is a targeted approach to induce immunity at mucosal sites via, for example, a nasal route.
So, all of our vaccines against SARS-CoV-2 are what I would call systemic vaccines. I'm guessing they don't produce much in the way of mucosal immunity. Do you think this is why there is so much breakthrough of virus in vaccinated individuals?
I think understanding what's happening in the breakthrough infections is really important. SARS-CoV-2 has been such an interesting experience for the scientific community because we've obtained and published data or released data in such a rapid fashion. And so it's much more well studied in the last year and a half than any virus probably has been in history. And so the uniqueness of the breakthrough infections, I think we've got to probably try to place in context with what we know about other infections and their breakthrough in vaccines. And thinking specifically about flu, we just don't monitor flu nearly as intensely as we've been monitoring SARS-CoV-2. So it's entirely possible that we have sort of similar levels of breakthrough infections, but we just don't catch them. I do think there are some unique features to SARS-CoV-2, though, that might facilitate a breakthrough in distinction to influenza viruses, for example. So the receptors that they use, where they're localized in the lung, and the polymorphisms and expression differences that occur in that receptor across the lifespan are things that I think people are still actively investigating. In terms of the vaccines themselves, while they're definitely not mucosally directed in the sense of like LAIV or the intranasal vaccines that Katherine was mentioning, they are quite robust. And so I would be surprised if there's at least some mucosal IgA, IgG, or at least a very rapid mobilization of those antibodies at that surface. I think it's a harder question whether or not there would be any T cells that would localize to the lung after those infections, but again, hopefully would be mobilized extremely quickly. And as Katherine mentioned, all of these questions are incredibly difficult in the human just because of the sampling issues. But there's been a number of studies this year that the investigators have released very, very quickly and preprint that at least are starting to get at some of these unique sampling modalities so that we can start looking at some of these responses in detail.
Interesting. So another area that I'm really interested in is immune evasion, and viruses are really good at this. Is immune evasion going to be covered very much in the meeting? And what are the latest advances in this area?
Yeah, so immune evasion is obviously a big problem in antiviral immunity, and previously has been extensively studied in HIV, influenza, And now we are closely watching how SARS-CoV-2 mutates and whether the new mutations not only evade the interferon responses, as Paul mentioned, but also what happens with antibodies, B cells, and T cell immunity. So far, the antibodies generated by COVID vaccine still recognize all the major mutations. So viral mutations and escape from adaptive immunity will be covered at the meeting. And the presentations will be showing the latest advances featuring large-scale analysis.
There's also an entire session on innate responses and the induction of cell death after viral infection, which is the other end of that immune evasion that Katherine was talking about. And then, you know, in the there are also going to be talks looking at how different hosts might have different susceptibilities to evasive maneuvers by the virus. So we know in certain high-risk hosts, the virus is much more fit in terms of completing its replication cycle than in low-risk hosts, and trying to find what features of that high-risk host biology are actually facilitating the virus and its replication cycle.
Oh, very interesting. So in addition to mechanisms, the meeting is also going to emphasize therapeutics. So what lines of research are leading to therapeutics against viral infections?
So the meeting is really looking at therapeutics from all angles. So we're all now incredibly familiar with the vaccines against SARS-CoV-2, but the meeting again is not just a SARS-CoV-2 meeting. So we're looking at you know, all types of viral targets for vaccination, and with the goal of improving vaccine efficiency, the ability of vaccines to induce broadly protective immunity against variable pathogens like influenza and SARS-CoV-2, and the ability to induce mucosal immune responses. And so obviously that vaccines as a platform for public health are far more potent than any other therapeutic will likely ever be. And so there's a lot of emphasis on that. But there are also talks on monoclonal antibodies, which again have been useful in SARS-CoV-2, but are also being applied in RSV and in neonatal RSV in particular. And then there are some interesting talks on small molecule discovery in targeting host factors. And this is a very exciting area because you know, host factor targeting is likely to be a much more difficult target for the virus to evade around. So there's no target in the virus itself that it can genetically mutate to no longer require. And so it's modulating the host, either immune response or the host factors that the virus co-opts to replicate to limit viral replication.
What are the latest tools and technologies that are driving new advances in the field?
So obviously Paul is a guru here and captures all the newest technology, but for me, it is really analysis of large data sets derived from RNA-seq technology, multi-parameter flow cytometry, sequencing T-cell, B-cell receptors all combined together to provide us a really in-depth understanding of immunity to viral infections.
Totally agree with that. I think, you know, the big data revolution really paid off in SARS-CoV-2 and the openness of the community and sharing that data well ahead of when you know, the normal publication cycle would have driven you to share it has you know, created advances much more quickly than we've had in any other pathogen. In the meeting, there's also going to be talks about advances in imaging and both intravital imaging and then organoid imaging in vitro that gets at some of the viral questions that are of great importance. And it's still, even with imaging, single cell RNA sequencing, and these other large data approaches, proteomics and so forth, it's the integration of all of those data and the computational analysis that's really key to moving the field forward, because it's getting easier and easier to collect data, and it's getting harder and harder to analyze data. And I think that's something that the meeting can really focus on, how collaborations among diverse groups can really move things forward much more quickly.
Yeah, I think that's really interesting. You know, these fields only go forward when new technologies come along to enable you to ask new questions. So it's really an important sort of component of a meeting like this. We've talked a lot about SARS-CoV-2, of course, because it's obviously a very hot topic, and your meeting is beautifully timed to address what's going on with the pandemic. So I want to ask you this question, and it's just your personal view. How do you see the SARS-CoV-2 pandemic unfolding over the next decade?
Well, for me, it's clear the SARS-CoV-2 pandemic will become endemic in years to come. And as the global coverage increases, we will be less and less susceptible to severe and fatal disease outcomes. COVID vaccination might be a part of our annual immunization regimen as SARS-CoV-2 mutates, or we need to boost our immune responses.
I agree with Catherine entirely. I think it's clear it'll become endemic, and it's a question of what that endemicity looks like, particularly since we don't have any experience with the other coronaviruses in terms of sort of chasing them with vaccines. We're putting the virus under a lot of pressure, which is a good thing, and it will be interesting to see how that immunity interacts with the virus over time. Based on the prior experience with coronaviruses, I think the hope would be that as we build up rounds and rounds of immunity, it becomes a common cold virus in hopefully relatively short order, but I don't think we'll ever be rid of it.
So this is just a question about the coronaviruses that infect humans. So if you just set aside MERS and SARS-CoV-1, there are 4 other coronaviruses that cause the common cold in humans. Are they possibly remnants of past pandemics that maybe happened decades, centuries ago?
So it's my understanding that there's quite strong evidence that OC43 is what's sometimes referred to as the Russian flu from 1883, maybe something like that. So yes, I think so. I'm sure they all were actually pandemics when they were introduced and maybe got conflated with influenza or one of the many other sort of historical reports of a disease that we don't really know what it was.
So a long and storied history then. Okay, do either of you have any sort of final comments that you'd like to make or anything you'd like to mention?
I'll just say, I think, as a community, the viral immunology community has performed a remarkable service for the world in the last 18 months. And, many of the leaders of those efforts that have led to the rapid development of these vaccines, rapid development of therapeutics, are going to be at the meeting. And I think for, again, all of us to come together and actually interact in a personal way is just so valuable at this time. Also we are sort of at this hopeful, dawn of a new era where maybe we have better control over the pandemic, and, at framing and asking the questions for, what did we do well, what did we do wrong, and what should we do next, I think is really critical. And then the last thing I'll say is, from a professional point of view, the people that have suffered the most over the last year and a half are our trainees, students, and postdocs. And it's so critical to them to have this chance to get back to doing science the way we used to do it, which is collaboration and conversation and sharing of data in a way that they haven't been able to do it. And I'm really hopeful that we can provide a lot of opportunities for those interactions to happen.
Yeah, and I'm sure this meeting will give them ample opportunity to do that. I think it will be a really wonderful meeting.
It was beautifully summarized by Paul. I just would like to say that we look forward to meeting everyone in Keystone.
Okay, well, thank you both for this fascinating discussion, and I look forward to hearing more in person at the meeting in January. Thank you very much.